thank you very much for the introduction. Sorry, guys, I was a little bit late because I had some technical problems with my computer and afterwards connecting to the Sky room. So without further ado, I start my presentation today about advanced biofabrication strategies for tissue engineering, regenerative medicine, and organ on a chip. As Majid mentioned, I'm an assistant professor in Institute of Biomedical Engineering in the Faculty of Medicine, University of Montreal, and my lab is located in the hospital, Sainte Justine Hospital, and I'm also affiliated to Montreal TransMedTech. I have a chair position in 3D bioprinting and regenerative medicine. So, if I want to talk a little bit about my background, I was born in Tehran, and as you can see, as you know, because I'm talking to most of the Persians here in this group, I usually used to go to the Houman summit and hike there and think about how I can contribute to during my the hiking, think about how I can contribute to the science and technology. And during my teenager time, I used to also buy this magazine called Danishmand, and it was a monthly magazine and journal. And reading all the content in one day, and then waiting for another month for the new issue. So, and you can see, I can assume that this journal is still continuing, and you also read the great articles in this journal. So, anyway, you can see my long journey from my bachelor. I did my bachelor and my master's in Iran, and then I worked around approximately about five years in industry, in pharmaceutical industry, in oil and gas industry, and then I moved to Canada, to University of Ottawa, and then worked as a research associate for a year on the fabrication of membranes for separating the biological solids and the biological wastes. And then I started my PhD at the University of Montreal, and mostly working on cardiovascular tissue engineering. And then I moved for my postdoc to University of Toronto, and I was working on cardiac tissue engineering. And then I got my position at the University of Montreal this year, and now I'm working, and I have my own lab, and we are developing. We are mostly working on cardiac tissue engineering, and this is my research interest. So, if I want to switch the gear and talk about tissue engineering, let me just hide this. Yes. This is Luke. Luke, at the age of 10, he was suffering from a neurodegenerative disease, and he had to undergo several surgeries, and he could not, enjoy the life. He could not play with his clothes, and he had problems. So this neurodegenerative problem led to bladder failure and kidney failure. So thanks to tissue engineering, he got a new bladder from his own cells, and now at the age of 27, in his college, he's a wrestling coach, and he enjoys life. And so, as you know, tissue failure is a tremendous clinical burden in the world. Approximately 5 million surgeries are done in the US to replace damaged tissue. This includes different tissues and organs, like orthopedic, cardiovascular, or internal organs. And the problem is that tissue transplant supply is insufficient, and synthetic. The one of the way to actually replace organs or damaged tissues is to replace it with the synthetic implants. But synthetic implants fail because of wear, fatigue, or by compatibility problems. So there is an unmet need for tissue engineered grafts and organs. So that's why the concept of tissue engineering has been developed fast during past decades. So if we want to look at the fundamental of tissue engineering, the basic of tissue engineering is just to get the biopsy from the patient and then digest the cells to the single cells and culture them in the petri dish in the lab, and then we combine these cells, the patient cells, with the scaffold that we made in the lab. This scaffold acts like a structure, initial structure for the cells, and then we matures these tissues by using chemical or mechanical or electrical stimulus. And then once the tissue is matured, we can implant it back and replace the damaged or diseased organ. There have been some successes in tissue engineering, like tissue engineering of bladder, urethra, ear, or other or trachea. So, and this is still continuing, and most of the work has been done in research labs, and now there are efforts to move forward to clinical applications. So during last decade, it has been like tremendous progress in tissue engineering by discovering novel chemistries or combining growth factors with these novel chemistries and the effect of mechanical properties of the scaffold on the cells, or using or in the engineering tissue architecture part, using the decellularized organ or self-assembly tissues or self-assembly structure, and more recently 3D printing or 3D bioprinting. And in the concept of engineering cells, it has been tremendous or significant progress of by discovery of the induced pluripotent stem cells, and then more recently CRISPR-Cas technology. So the more recent advancement in tissue engineering is tissue vascularization, because once you have the thick tissue to integrate this thick tissue to the body or to the adjacent or neighboring organs, you need to connect the artificial tissue to the body. So the vascularization is the most important part for the integration to the host. So now I want to a little bit switch the gears. Tissue engineering not only be used in tissue replacement. More recently, it has been used for in drug discovery and screening pipeline. So as you know, like for any drug to come to the market, it takes around 10 to 15 years, going through this pipeline from basic research in the lab, in vitro studies, in vivo studies, and then clinical studies, phase 1, phase 2, and phase 3, and then FDA approval. Pharmaceutical companies spend billions of dollars in R&D to bring new drugs into the market. However, some of these drugs, once they go through these pipelines and get to the market, they fail after that, even after getting to the market, mostly because of the efficacy problem and efficacy and safety problem. So the most famous case was YX, that was a drug for arthritis and osteoarthritis that led to cardiac toxicity. So the company Merck had to pay billions of dollars in compensation to the patients and to the family of the patients who lost their life because of this drug. So it seems that this pipeline, like there is a problem during the in vitro or in vivo experiments that made this problem to happen. So this is more because mostly using animal models in this pipeline. So once we want to test it, or discover new drug, we use animals that are not really mimicking the human physiology or pathophysiology of human body. So, or we use 2D cell culture. So both of these models cannot really mimic what we have in our in the human body. So this leads to actually the need for having a new model that recently have been developed. We call it organ-on-a-chip model or 3D in vitro models that can be used in drug discovery and screening application. So these models are really like inexpensive, cheap, and you have the control, and they are really high throughput, and then we can really mimic the structure and function of tissue in a miniaturized way. So combining these three models, animal models or in vivo models with organ-on-a-chip models and in silico models, computational models, can be really helpful to have better understanding and better understanding of drug effects in human body. So more recently, the concepts of human on a chip and also has been introduced by the Weiss Institute in Harvard University. So the concept is that uh, is to combine different organ on a chip models and then see the effect of the drug on the upstream and downstream of the tissue. So this is really cool, and so this is kind of the introduction that I had about tissue engineering. Now I want to switch the gear again to some examples that I have been done during my research journey, from my PhD, from my postdoc, and from my collaborative work about tissue engineering and organ on a chip. So the first project that I want to talk about is fabricating the vascular graft using electrospinning technique, and combine these electrospun fibers with bioactive coating to make a small diameter vascular graft. So a little bit of background. So as you know, the cardiovascular disease is the leading cause of death worldwide, and this is mostly because of the atherosclerosis. That atherosclerosis is basically is depositing the cholesterol, fatty stuff on the wall of the coronary arteries and in the heart, and then blocking the flow, and that leads to myocardial infarction or heart attack. So the trap is that in the earliest stage of the disease, the first step is to to do angioplasty and the stenting to open the blood coronary artery. However, in the later stage, when the arteries are really closed and blocked, the gold standard is bypass graft using the saphenous vein from the patient. So the surgeon will replace the coronary or blocked coronary artery with the saphenous vein from the patient. But most of the patients cannot provide this saphenous vein because of the previous disease or surgery that they have. So they had. So the alternative or another option is using the prosthetic or artificial vascular graft. So, but most of the time now, when you look at the companies or commercialized vascular graft in the market, most of them have a large diameter, more than 6 millimeter. However, the coronary arteries have a small diameter, less than 6 millimeter. And the material that usually company use to make this vascular graft is Teflon and Dacron, that we call it PT or PTFE. So there is as I mentioned, there are clinical limitations for using these materials, PTFE or Dacron. These materials are really stiff. Their mechanical property cannot match the mechanical property of coronary artery. So there is a compliance mismatch between these two materials. I mean, with the coronary artery. And the other thing is that the surface of these materials are inert. So the cells they don't have affinity to attach and to proliferate on these materials. So basically, because we use these materials as a coronary artery, the endothelial cells cannot attach on the surface of these materials. So there is an inadequate endothelialization that leads to lack of patency. Patency means openness of the artery, and then later on to the intimal hyperplasia. So, but if you look at any tissue from our body under the microscope, you can see this fibrous structure, the extracellular matrix, and the cell. So one of the way and one of the versatile way or technique to mimic this structure, this fibrous structure, is electrospinning. And so in this project, the objective of the project was to develop a small diameter vascular graft, which are compliant, which have mechanical property close to the coronary artery, and also cell inductive, so the cells can attach to the surface, and so kind of addressing this clinical limitation that we have. So by that, this work was done in University of Montreal, Polytechnic of Montreal, and the CR Houman and the Research Center of University Hospital of University of Montreal. So as you can see, our arteries contain three layers. The first layer is intima, that is exposed to the blood and contains endothelial cells, and then the second layer is media in the middle of the coronary artery, that contains smooth muscle cells, and the outmost layer is adventitia, that contains more collagen and fibroblasts. So with electrospinning, I don't think that I need to explain electrospinning, but this is the technology that was rediscovered from the textile industry to make fibers. And So the technique is very simple. You put the polymer solution inside the syringe, and then by adjusting the parameter like the voltage, the distance from the needle to the mandrel, the mandrel speed, and the polymer flow rate, and all these, and the viscosity or rheology of the polymer. You can have different fibers like aligned fibers or random fibers with different structure and porosity. So, so we did the same thing. So we developed aligned or random fibers for the first layer, for the intimal luminal layer, and then and we use backbone or PET. So since the surface of this material is inert, so we use plasma polymerization to kind of coat them with the primary amine group, and then we grafted chondroitin sulfate. Chondroitin sulfate is a sugar, is one of the components of our ECM or extracellular matrix. It's biodegradable, biocompatible. So we added chondroitin sulfate, and then we studied the effect of cell adhesion, proliferation in a static culture, and also we exposed these cells to the shear stress similar to the shear stress in our coronary artery in a dynamic environment. So as you can see, we were able to successfully fabricate random fibers, aligned fibers with the porosity around 87% for diameter of for the random fibers 3.5 micron, and the fiber diameter around 500 micron. So as you can see, we see that the endothelial cells on these surfaces, and endothelial cells form or cover all the surface and form a monolayer on the top of the surface. This is really like suitable for this kind of application, because as you can see in the woven PET that has been used, that is commercially developed and is being used by the surgeon to replace coronary artery, the pores are really large and the cells go inside the structure, so they cannot form the monolayer that is really mimicking the structure of the coronary artery, the first layer. So you should have a monolayer of endothelial cells. So the pore size of our fibers are really small, so the endothelial cells cannot infiltrate inside the structure. So this is really suitable for our application. And then once we see the endothelial cells, we study the adhesion of the cells, adhesion of the cells. Once the scaffolds are coated with the plasma polymerized coating and the chondroitin sulfate. So what we saw that the cells are really happy. They attach in the day one, and they grow through the culture time till after 21 days. And on the bioactivated surfaces, they really like bioactivated coating. They really promoted cell adhesion and growth. And this was similar for random and aligned structures. So the cells also responded to the structure. So on the aligned fibers, they are really spread towards the direction of the fibers, and then on the random structures, they're really like randomly oriented or spread. So the cells are happy. They form the focal adhesion on the material. So it means that they're attached, and they also communicate with each other by expressing the biomarkers here that we show with in white. So And once we expose these cells to shear stress around 15 dyne per square centimeter, which mimics the physiological shear stress, we saw that the cells on the bioactive coated scaffold can really retain to 90% of the cells can be retained compared to the control sample without coating. So only 30% of the cells or 25% of the cells can be retained on the control sample compared to the bioactive coated sample. So it means that the bioactive coating can really promote cell retention once they're exposed to shear stress. So this is the really like the project was mimicking the first layer or luminal layer of the vascular graft, and then we move forward to mimic or develop the second layer, which is the medial layer, contains circumferentially aligned fibers. So we really need to have aligned fibers, and so we were able to successfully develop these fibers with 87% of porosity and with the pore diameter of 6.5 micron and average fiber diameter of 900 nanometers. So basically, like as we use the same material, we did the same process of coating with primary amine and then chondroitin sulfide. This time to further promote the bioactivity of the scaffold, we kind of grafted epithelial growth factors in an oriented manner on the surface of the chondroitin chondroitin sulfate by NHS chemistry to improve the growth factor potency scaffold bioactivity. And then we see that the vascular smooth muscle cells, and we studied the cell communication with the scaffold. So as you can see, the cells are really like the bioactivity or the growth has been promoted on the surface with the growth factor immobilized scaffold compared to the control sample, and this was really significant to the different layers of the scaffold that you have only primary amine, you have only the chondroitin sulfate, and you have the growth factor on top of that. So the adhesion really didn't change, but the growth has been changed a lot. So the only difference here is that we made fibers with the larger pore diameter. So this was intentional because we wanted the cells, because here in the medial layer, the cells can really should infiltrate inside the scaffold. So here in this histology image, you can see that the cells really infiltrate and penetrate inside the scaffold. So since you have the large pore size, the bioactive coating, including the plasma, chondroitin sulfate, and the growth factor that they can penetrate inside the scaffold, and the cells continue or follow them, and then the cells also go inside the pores. And so this was really the summary of this project. So we made this two-layer vascular graft, small diameter, less than 6 millimeter, to really address the clinical limitation that I mentioned in the beginning, like compliance mismatch, mechanical property mismatch with the coronary artery, and inadequate endothelialization. So so now I wanna switch the gear to another project. So once you make so by answering, so we have to answer this question really to get to this project. Why is there a lack of adequate in vitro engineered tissue? Why we have we don't have enough in vitro engineered tissue. So the answer is that lack of vascularization. Vascularization, as I mentioned, is the really important concept engineered tissue to be integrated to the host tissue, right? So this work also has been done was done in University of Montreal and Polytechnic of Montreal. So as I mentioned, that this vascularization can be done by one of the way that we can do this vascularization is pre-vascularization to accelerate functional connection with the host tissue after implantation. So to further study this, we actually developed the micro strands, and then we immobilize VEGF or vascular endothelial growth factor on the top of the heparin sulfate on the scaffolds on the micro strands, and then we wanted to actually study the angiogenesis or blood vessel formation in the culture. What we have seen is that, or we observe, is that the cells, after being in the culture, the endothelial cells started to sprout from the strands and make our tissue vascularized. So we have the microvessel formation. So, but the only challenge is that, okay, so you can have angiogenesis or vascularization in the thin tissue because endothelial cells, they do their job. The endothelial cells tend to make these microvessels or tend to promote angiogenesis in contact to the growth factors, for example, VEGF, EGF, or other growth factors. But this is really for thin tissues. These blood vessels or microvessels here are not perfusable. They are not open. They don't have open lumens. So that's a really problem for thick tissues when you have thick tissues. So how about when you have the thick tissue? So one of the solutions is to actually build in vascularization in the thick tissue, or building blood vessels in the thick tissue. So one of the projects that I work is that to make kind of micro vessel on a chip. This was done in University of Toronto. So the really, like, the objective was to design and fabricate an integrated microvasculature on a chip for assessing dynamic events. What does it mean? So we use 3D printing. So we had a bioelastomer that was really difficult to 3D print. So we used the fresh 3D bioprinting technique to print our bioelastomer. So fresh technique, I think you know, that is a really interesting technique that recently developed by the researchers in Carnegie Mellon University. In this technique, you have hydrogel, a secondary hydrogel, and then you print your own hydrogel or elastomer inside that hydrogel, and then you cross-link. Since our elastomer was photo-cross-linkable, we can cross-link these tubes, and then we assemble these tubes on the 96 cell plate. And as you can see in this image, you have the microvessels or microvasculatures on chip. The purpose is that once you have this, it means that you have one microvessel or microvasculature, so you can form different tissues around it. You can form cardiac tissue, liver tissue, cancer tissue, or any other tissue around these vasculature. And these vasculatures are really permeable and perfusable. So it has open lumen, and it has the connection to the outside, because it has really process structure. The wall has process structure. So even, like when you have different tissues, you can connect different tissues on a 96 well plate with this microvasculature. So I just want to show you some of the images that we have from the wall of these vasculatures. This is the microvasculatures with around 500 micron diameter, and you can see really like the microstructure of this this blood vessel or microvasculature is really really porous, so it really like promotes transferring oxygen, nutrients, and all the stuff from inside out or outside in. And so we also endothelialize this microvasculature, so the endothelial cells can form the really like monolayer, and later on we form the vascular cardiac tissue on around this microvasculature. As you can see, the cardiac tissue now is here is beating, and so the cells are happy. So this was like maybe 2-3 weeks in culture. So and every 2 days you change the media for the cardiac tissue, and then you change also the media for endothelial. So this is really like cool stuff. And then in another project with the collaboration with University of Victoria, we kind of like we wanted to develop a technology using microfluidic printing to make hollow fibers, or even like the fibers with functional tissues that they have biophysical or biochemical features for tissue engineering application. This was the work, as I mentioned, that was done by in collaboration with the University of Victoria. So we made this kind of like 3D printed a mold, and then we printed these fibers with different features, as you can see, with different cross section, like circular, or even like a cross, hollow fiber, or the fibers that are really like the loop fibers, and then you can have different like knitted, woven, or different fibers, different structures. And in this SEM, you can see that, okay, you can have different features, kind of micro grooves inside and outside the fibers. So you can really control your fiber structure, single fiber structure, or macro structure of the hydrogel. Here we use alginate to make these fibers. So this is a concept that's a microfluidic printing that is really like recently developed, and to make different fibers. So, and we also evaluated the cell activity on these fibers. As you can see in these confocal images, that the cells are really responding to the microgrooves. For example, here on the grooved structures, they're aligned towards the groove. On the plane structure without grooves, they really like cover the surface without any alignments. And the cardiac cells, these are the brain cells, these are the cardiac cells, have the same phenomena or same response. So really like the objective was making these fibers using this novel microfluidic printing technology. So from this, now this project is ongoing, so we can have different patches or different like woven structure for skin tissue engineering, cardiac tissue engineering, or other application that really, I mean, these fibers are suitable for. So now I want to change the gear again to another project. So in all these projects in cardiac tissue engineering, specifically the project related to the heart, we use cardiomyocyte. Cardiomyocyte is really like the cardiac muscle cells or muscle cells in the heart that they do the job. So they really like once I mean most the function of the heart is really like dependent to the cardiac myocyte. Cardiac myocyte they really like produce forces and pressure, so they can really like push the blood out of the heart. So really like to by the discovery of induced pluripotent stem cells, now it's possible to make any kind of cells from the patients, and we could have the personalized or precision medicine. So in the lab in University of Toronto, I have been involved in differentiation and sorting of these cells for tissue engineering or regenerative medicine application. So what we did is that we get the cells from the skin, my skin or my colleague's skin or any other person's skin, and then we kind of program or reprogram these cells to the pluripotency stage, and then by differentiating these cells using different growth factors and the specific protocol, you can get these cardiomyocytes that they are building on the on the petri dish, right? So the problem is that okay, once we differentiate these cardiomyocytes, the differentiation yield or performance is not high. So you have cardiomyocyte, you have cardiac fibroblasts, you have endothelial cells, smooth muscle cells. You have a heterogeneous population of the cells, right? So this heterogeneous population is problematic. For example, in tissue engineering or regenerative medicine, you really need to have a pure or kind of almost pure population of the cells. And like the conventional technique is using flow cytometry, FACS or MAX, magnetic flow cytometry or flow cytometry to kind of sort or separate these cells from each other. But the thing is that there are not a lot of biomarkers, because these techniques are really depend on the biomarkers. So you need to kind of tag cardiomyocyte with a biomarker and then separate them based on their based on these biomarkers. So there are no specific biomarkers for cardiomyocyte. Recently, there was a discovery of a SIRPRA surface marker for cardiomyocyte, but this biomarker is not really specific. So what, like the other technique, the most, I mean, most of the labs, they don't use FACS or MAX flow cytometry to separate or to sort this kind of cells. They use pre-plating. Pre-plating is really like a very basic technique to do that, because you kind of the cardiomyocyte cells are not adherent cells. Once you plate them, they stay on in the media, and the endothelial cell, fibroblasts, smooth muscle cells, they attach to the petri dish or to your flask. So later on, after 1 hour, you can separate them. Still, after pre-plating, you don't have a pure or really like homogeneous population of the cardiomyocyte. You have some impurity of other cells. So the technique is not really specific, and it's really time consuming. You have, for each stage of pre-plating, you have to wait 1 hour. And if you do 2 rounds of pre-plating, you take 80%, you get 80% of cardiomyocyte. And it's not consistent. Every time you do this, you get different population of cells. So with the collaboration with the University Technology Sydney, so they developed this spiral microfluidic platform. They use this platform for sorting or separating the CTC or circulating tumor cells. So we find out that, okay, maybe you can use these cells to sort the cardiomyocytes from other cells without any labeling them, or and also we can speed up the process. So what we did is that we this spiral microfluidic is like a centrifuge. So it has a entrance and then two outlets. One outlet it really works. It really like separate the cells by their physical differences, by their sizes. So the cardiomyocyte and non-cardiomyocyte cells can be separated from the two different outlets by, different sizes that they have. So what we show is that, for example, once we use the initial cells of 70% or 60%, 65% cardiomyocyte in the inlet, in the outlet you can get more than 90% of the purity of the cardiomyocyte. So, and then you can make the tissue from these cardiac cells, and you can see the cells are really happy beating, and they have all the features of cardiomyocyte. So you can also connect these. So this is one-time separation or sorting or enriching. You can connect two different or two, three of these spiral microfluidic together in series, and you can get more than, close to 100% cardiomyocyte purity. So So once we have these cells, we can use these cells in different platforms to kind of mimic the structure of the heart, or make cardiac patches, or other. So what we use these cells to make a cardiac left ventricle. So in this project, again in University of Toronto, we try to use microfabrication technology, here soft lithography, to kind of simulate or mimic the structure of the left ventricle. If you look at this picture on the top, right, you can see that really like our heart contains fibers that are they change their angles from the apex, from the bottom of the heart to the top of the heart. So these angles, like change from -60 to +60. So we wanted to mimic this structure by using soft lithography. This is the procedure that we use to make these channels inside the scaffold, and the scaffold is really porous scaffold with the micro channels on it, so we can kind of have a contact guidance for the cardiomyocyte, and later on we can kind of like roll up these scaffolds on the mandrel, and you could have a mini heart. So as you can see here, so we have really like a structured cardiomyocyte, and that they are beating here in this movie, if it's clear, and they have we measured their beating pace, and so so this project also is ongoing project. So this platform can be used for drug discovery and the screening later on, or I mean, since you have the mini heart, you can also mimic the disease, heart disease like cardiac fibrosis, hypertension, or other, by using specific cells, patient-specific cells. So so you can have really like different platforms. So in general, I just want to conclude my talk is that, so during my talk you saw, or you kind of appreciated different biofabrication techniques, from electrospinning, microfabrication, 3D printing, microfluidic printing, even like cell sorting. So these advances in biofabrication technology can lead to have the complex biological structure with integrated vasculature And then, so the only problem is that the successful clinical application of this engineered tissue is limited. So, but as I mentioned, by the recent innovation in stem cells, genetic editing, immunomodulation, and 3D biofabrication, we can boost and promote the clinical translation of engineered tissue. So a little bit about my future work and the work that we are doing in our lab. Our vision are really like to combine functional biomaterials with advanced biofabrication technologies, like the technologies that I mentioned: 3D printing, bioprinting, microfabrication, electrospinning, and other advanced biofabrication technology to make functional tissue engineered constructs for organ on a chip and drug discovery application and clinical application. So in this regard, we are now focusing on making or mimicking heart muscles or heart using 3D bioprinting technology to make the cardiac patches or to make the platforms for or mini heart for drug discovery application, and these cardiac patches for implantation, or heart valve using 3D bioprinting technology, mini heart valves or real heart valves for implantation. So really like a multidisciplinary project. I have several collaborators who are clinicians and in the hospital. So now we are working on these kind of projects. So and the other project that I'm really interested because I'm kind of I have been working with biomaterials for a long time is to make the immunomodulatory coatings for medical devices uh, inspired by the body's inherent molecular defense. So we get the idea of our body system or immune response, and we try to get those molecules and we kind of integrate them inside the or inside the polymer chain or biomaterials chain in the lab. So, and then we can have the different coatings for medical devices. And also, as I mentioned, I'm interested in the organ on a chip technology. We can have different organs. For example, one example here is the human intestine that we are working on. And also surface patterning is another interest that we have in the lab. And so at the end, I would like to really thank my external collaborators from different universities, from different countries, Australia, United States, Canada, and also the funding sources from the government of Canada and government of Quebec, and also my school, University of Montreal, TransMedTech Institute, and Sainte Justine Hospital, who really like trusted me and in my research, and they funded my research. And I would like also to thank all my mentors from the beginning of my career, academic career, which was very long, and they really like contributed to my success. So by that, I would like to thank everybody, and I'm happy to answer your questions.